In the last video, we looked at what actually goes on inside a eukaryotic cell, focusing mostly on a generalised animal cell. Now the thing is, eukaryotes are kind of in a minority. Most of the life on this planet is something else entirely. Prokaryotes. Now then, if you want to know what the most metal organisms on the planet are, you're in the right place. And I'll tell you now, it's bacteria. They can survive and thrive in conditions where no other living thing can. Now there are species that can even survive exposure to hard vacuum. Okay, they sporulate and basically just go into hibernation. But there are other ones that can live comfortably in water at temperatures of over 120 degrees C. So boiling water. So let's have a look inside these things then, shall we? First thing we're going to look at is the structure that's kind of analogous to a eukaryotic nucleus, the nucleoid. Now the nucleoid is, well, it's not a regularly shaped thing. It's not sort of bound in my memories or anything like that. It's just a region within the cell of a prokaryote that contains the primary DNA. It's also got in there with it proteins and enzymes to transcribe DNA and RNA and help out with cell growth and development. So generally you're looking at sort of 60% DNA, about 30% RNA and about 10% protein at any one time. Protein, you're looking at things like, well, but basically your transcription and translation enzymes and your DNAs and all that sort of stuff. Yeah? Right. Since we're talking about DNA, we'll move over a bit further into the cell, and you see those things there? Those things are plasmids. Now, plasmids are small circular structures usually of, and this is important, self-replicating genetic material. Now, they tend to be somewhere between 1,000 and 200,000 base pairs in length, so pretty small. And they are capable of moving from cell to cell on their own. And some people call them jumping genes. Now, they usually code for a small number of genes that can kind of lend the cell they inhabit a specific characteristic. I mean, best known example of plasmids are the ones that are involved in antibiotic resistance in bacteria. Now, the term plasmid goes back to 1952, when an American molecular biologist by the name of Joshua Lederberg originally used it to mean any source of genetic material in a bacteria from outside its chromosome, well, including viruses. That was modified in 1968 when that definition was refined, basically to say that not viruses, because these plasmids have got to be able to replicate themselves and spend most, if not all of their time outside the chromosome of the host cell. Some, I say most because some plasmids do actually integrate themselves into the genome of the bacteria in order to replicate. So what they do is they use restriction enzymes to cut the DNA. They'll insert themselves, anneal the ends, get replicated, and then use the restriction enzymes again to kind of lift themselves back out. Pretty clever, eh? Others can self-replicate. Don't even need to do stuff like that. Now, since they do stuff like that, molecular biologists have cottoned onto it and kind of started making their own artificial plasmids. And these things, well, they use as vectors in molecular cloning. Now, I don't mean basically making new sheep. Molecular cloning, what we're talking about there is putting genes into other genomes. That's what cloning actually is. Now, these artificial plasmids are driving the replication of recombinant DNA sequences within host organisms. So you can make, for example, look at E. coli produced penicillin, for instance. They did that using plasmids, and so on. Now, you can introduce them into a cell via things like, well, they call it transformation. Like viruses, you don't necessarily call plasmids life. I mean, viruses aren't life because they can't sense their environment, they can't reproduce on their own, and so on. Plasmids can reproduce on their own, but they can't sense their environment, and so on. Now, they get from one bacteria to another through conjugation, which is kind of like sexual reproduction in bacteria, except there's no sexes or genders in bacteria. We've just got a plus and a negative. A plus having the pili to sort of inject, and the -1 not having that. Interestingly, some plasmids actually carry coding to make them able to produce the sex pili. So that means they can basically make themselves and transfer themselves. Pretty good, that, isn't it? Let's have a look at ribosomes then. Now, you've encountered ribosomes at the last video. You know what they do. They build proteins. Now, there's two types. In eukaryotes, you've got 80S ribosomes. And in prokaryotes, you've got 70S ribosomes. Now then, what the hell does that all mean? Well, first, what we've got to do is actually look at the ribosomes themselves. What they do, how they're constructed, and so on. Now, what they do is they translate the code from DNA into proteins by stringing together amino acids in a sequence that is defined by RNA that's been transcribed from the coding strand of the DNA. It's all right, we'll do that in another video, and we'll talk about tRNA, mRNA, all that sort of stuff. All you need to really know at the moment is that ribosomes make proteins. Now, ribosomes are actually built of complexes made of RNA and proteins. And they are divided into two subunits. You've got a smaller subunit, which binds to the larger one, and the mRNA pattern kind of clicks through it. You know, kind of like one of those label makers. You know, you click the letter and you wind it on, you click, wind on, click, wind on, and you start, and you've got the word starting to build up on the bit that comes out. It's like that. Now, the larger subunit binds to the tRNA and the amino acids and the smaller subunit. So basically you've got like your DNA strand, sort of like that. You've got your large subunit, your small subunit, click. And then you have your protein strand sort of winding out of there. Well, like I said, we'll do more on that when we get to transcription and translation. Now, these subunits is where the 70 and 80 stuff comes from. Now, in prokaryotes, the two subunits that make up the ribosome are a small one at 30S and a large one at 50S. Now, yeah, I know, that adds up to 80, I know. But it's 70, okay? Because the small subunit has got a 16S RNA subunit, which is made of 1,540 nucleotides bound to 21 proteins. The large subunit is made of a 5S RNA subunit, which contains 120 nucleotides, a 23S RNA subunit, which is made of 2,900 nucleotides, and 31 proteins. I know. Don't worry, you don't really need to remember all that. I mean, I'm reading it off a script. Interestingly, ribosomes from bacteria, archaebacteria, which are absolutely ancient organisms that have been around for like billions of years, and eukaryotes are actually incredibly similar in terms of structure and makeup and everything. Now that kind of points to them having a common origin. The differences are in size, sequence, structure, and ratio of protein to RNA. And incidentally, it's these differences that allow various different sorts of antibiotics to kill bacteria, because what they do is they inhibit a particular type of ribosome, the 70S ones, while leaving the 80S ones completely unaffected. Here's something else interesting about bacterial and archaebacterial ribosomes. For just those, more than one ribosome can move along a single mRNA chain at one time. Meaning, you can, if you are an archaebacteria or just an ordinary bacteria, you can produce the finished proteins a lot faster than eukaryotes are capable of. I mean, you look at E. coli, for instance. It can go through an entire replication cycle in 20 minutes. You know, from interface to interface. For a human epithelial cell, you're looking at over an hour. And that's the reason. It's not just that they've got a smaller genome. Their ribosomes can work as teams. Now, going out to the edge of the bacteria, we've got a structure called the capsule. Now, the capsule occurs in a lot of different types of bacteria, but not all of them. What it's made up of is a load of polysaccharides. Except, I mean, there are exceptions to this, such as Bacillus anthracis, which is anthrax, has got a capsule made up of polypeptide D-glutamic acid. And E. coli, its capsule contains peptidoglycan and muramic acid. Most stains won't get taken up by the capsule, which means you're actually on the first stage of identifying the bacteria right there. Because if it's got a capsule, what you do is you end up using India ink staining, which leaves you with like a black field and then clear halos around the bacteria. Manival capsule stain, which again you got a clear halo, but with a bacteria stained pink on the middle and you got a bluey grey background. Or you could use the Kvellung reaction. Now, Kvellung is German for swelling. Now, this whole thing was devised in 1902 by a German chap by the name of Fred Neufeld, also called Neufeld's reaction. What it does is it uses antibodies to attach to the capsule, which make the bacteria appear to sort of swell up. Quelling is German for swelling. Now, so these sort of bacteria appear to swell up as this big sort of antibody capsule complex forms. And that increases the surface tension in the water around it and creates a clear outline around where the capsule is. So you can spot it pretty easily after that. Now, if you are a bacteria, a capsule is a very useful thing to have. For one thing, it protects you from phagocytosis by macrophages once you have invaded a new host, since the immune response will actually need an antibody specific to that particular capsule to attract any macrophages. And it also protects the bacteria from desiccation because it's got water in it. And it'll stop detergents getting in and destroying the lipids. See? Very useful thing to have. Also known in some cases as a slime layer. Now, that all sits on top of the cell wall. And in terms of outer casings, the bacterial one is the business, really. Now, we already know that plant cell walls are made of cellulose and fungal cell walls are made of chitin, which incidentally is the same stuff that the exoskeletons of insects are made of. So bacterial cells are pretty damn tough. In fact, the bacterial ace in the hole is peptidoglycan. It's a polymer made up of polypeptide chains cross-linked with unusual amino acids, such as, for example, alanine. Now, there's two types of cell wall you'll find in a bacteria, and they've got varying amounts of peptidoglycan. You can distinguish between them using the Gram stain, which I'll talk about later on in this video. And because of this thing, they're called gram positive and gram negative. Now, gram positive bacteria are stained dark purple by the stain and have got a thick cell wall made up of layers of peptidoglycan and molecules called teichoic acids. Now, these teichoic acids make the cell walls more rigid by attracting magnesium and sodium ions. Basically, they weld armor plate into the cell walls. Now, you see, this is why the Clostridium genus is the most metal of all living things on this planet. Because not only do they have metal in their cell walls, they will survive just about any conditions you put them into. You can boil them, you can chuck them in acid, you can send them out into space. They'll survive. See? Like I said, metal. Anyway, and by comparison, your gram-negative bacteria are pretty much wearing a tinfoil coat dipped in lard. With their cell walls, you've got a few layers of peptidoglycan surrounded by a second lipid membrane containing lipopolysaccharides, which, okay, you can actually find them in snake venoms, and they are pretty metal because snake venom actually liquefies cells. And also in there, you've also got your lipoproteins. Now, fortunately for us, at least in terms of pathogenic bacteria, most of them are gram-negative, so they're pretty easy to kill compared to the gram-positive ones anyway. Incidentally, most antibiotics actually work by damaging the structure of the cell wall. Oh, and yeah, if you want damage to the cell wall, wait till I get on to talking to you in a later video about the immune system, and in particular, the complement system. Let's put it this way. Things get pretty violent, at least on a biochemical level anyway. Now, do we need to look at anything else? Yeah, we do. I did promise you we'd talk about a Gram stain. Now, the Gram stain was developed by a Danish bacteriologist by the name of Hans Christian Gram. And I just hope he wasn't as bad a house guest as Hans Christian Andersen apparently was. Look it up. I think he was stopping with Arthur Conan Doyle, and put it this way, he didn't make any friends. Yeah, so Hans Christian Gram, 1884, came up with the Gram stain. He was working with a chap by the name of Karl Friedlander in the mortuary at Berlin City Hospital. And what he was trying to do is find a way of making bacteria more visible in stained lung tissue sections. Now, apparently, as it says in the paper, typhus bacillus did not retain the stains that he used. So, in other words, what we're saying is typhus, so Rickettsia species, are gram-negative. So really, he'd kind of done the first bit of using his stain for identification without even realising it. So how do you actually do this Gram stain then? First off, what you do is, you get your slide and you smear some bacteria on it. And then you got your Bunsen burner and you flame it. So you just like whip it through the flame to heat fix it. Then you give it a chance to cool down. because you don't want to be putting cold liquids onto hot glass, because what you do then, over a sink, and be careful not to get this stuff on you, because it's called a stain for a reason, you add the primary stain. So you pour a load of oxalate crystal violet over it. Now, oxalate crystal violet is dark purple. And yeah, it's going to stain your fingers. It looks a bit like, you know when you get a biro and you rip open the tube with the ink in it, and the ink's got that sort of like a pearlescent kind of look to it. That's what oxalate crystal violet looks like, except it's not as thick and sticky. Well, not quite, anyway. So once you've got that on, you leave it on for usually about 30 seconds. Then you wash it off with water and pour on some grams iodine. Now, grams iodine, a little bit different. It's a mixture of iodine solution and potassium iodide. You give that about a minute. Now, the reason you do that is it makes things more visible, because the crystal violet iodine complex is a bigger molecule, which you will find out is important later as well, and it's also insoluble in water. So yeah, like I said, you give that about a minute, then you wash the slide down with acetone just for a few seconds, and then you rinse it straight off. Now what that does is it decolorizes the sample. Now that works by dehydrating the peptidoglycan, which then makes it shrink. And it traps that crystal violet iodine complex inside the cell wall, because now it can't get out anymore because it's too big. And you've shrunk the cell wall down around it. Now, in terms of the gram-negative bacteria, that same treatment damages the outside membrane, allowing that complex to be washed away. So that means that dark purple only stays in the gram-positive bacteria. Yeah? Now then, so you can see the gram-positive ones now, but you can't see the gram-negative ones. So what you do is you add a counter stain, such as, for example, carbol-fuchsin. Now that stains your gram-negative cells rather a nice shade of pink. So that means when you look down your microscope, you can say that, for example, you can see gram-positive bacilli, which are brick-shaped, probably anthrax. See, you can identify just by doing all that and looking down a microscope. Or another possibility might be, if you are working in a hospital, you're seeing gram-positive cocci in chains, streptococcus. Probably this person this came from has probably got a sore throat. And you can do that before you get any biochemistry going. I mean, you could even, from that, start looking at prescribing antibiotics. Because streps, you know what they're susceptible to. You can then sort of send information up to the ward saying, right, well, they've got this, so if you put them on this, it'll sort it out. Right, okay then. Summing it all up then, what have we learned from this video? First off, we now know the structure and functions of various parts of prokaryotic cells. We also know how to carry out a gram stain and how it all works. Now, before you go, remember to check out the Heavy Metal Science Teacher Facebook page and Twitter feed, where you're going to get news of any new videos that I put up. You'll also be able to ask about the subjects for future videos, but remember, keep them science. Links for those are below in the comments.